Hey, what's up guys? BIOBEAN here. Today we're going to be talking about the Human Genome Project, so let's get started. So the Human Genome Project was started in 1990. This was done with the goal of trying to understand what the sequence was of nucleotides within the human genome. So what they did was they took the genome from a variety of individuals and they attempted to sequence it. So the reason they took it from a variety of individuals is because every person's genome is unique to them. That's how, if you leave your DNA at a crime scene, we are able to tell that is your DNA because there's particular markers that are unique to you. So they took the DNA from a variety of different people to kind of create a better average genome. So to look at what would be common across different people to kind of get a standard genome. So what they did was they took those genomes from a variety of different people and they wanted to sequence the haploid genomes. They only wanted to look at a single copy of each chromosome and sequence that. Normally humans have a diploid chromosome, but they wanted to just kind of get a baseline, what is the sequence of each individual chromosome, not really concerning about whether it was from the mother or father. And so they were sequencing the entire genome, including the Y chromosome. So this was because they took all of the genomes from male individuals, and so that genome included that Y chromosome. So the haploid genome with the Y chromosome is about 3 billion base pairs. So that was an immense undertaking in the 90s, as we had relatively no technology surrounding sequencing. And because the Human Genome Project is really why we have such cheap sequencing today. So if you know of 23andMe or any of the other kind of genome projects that are catered towards individual people, those only exist because of what was done in the Human Genome Project and the undertaking that they did in order to develop better sequencing abilities and also to better understand what the human genome looks like, with the hope of one day being able to do better science that involves the whole genome, as well as being able to tailor medicine towards individual people according to their genome. The Human Genome Project was declared finished in 2003, where they had sequenced the entire human genome. We have continued to add to this repository, sequencing more and more individuals, and at this point, we have a database that has hundreds of thousands of genomes present and is constantly being added to every single day. So I said the Human Genome Project was finished in 2003, so let's get into what they found to be within our genome. When the Human Genome Project was announced as complete in 2003, they suggested that only 2% of our DNA actually was essential, that it coded for something. So they found that only 2% of our DNA actually coded for genes that would go on to make proteins. So the rest of the DNA, all other 98% of it, was considered to be useless DNA. So that might seem like a little bit of a hubris when you say that, well, only 2% we found as being genes, and therefore all other pieces of DNA, because we don't know their function, must be useless DNA. It must be junk. So it seems a little outrageous to say, but they needed kind of a big headline. They just spent like a decade and billions of dollars, or maybe millions of dollars, investing in this project, and so they needed a big headline. Most of your DNA is junk, and only 2% actually codes for genes. But then, since then, we've done a bit more digging into the genome, and we've come to understand a little bit more about what is in our DNA. All right, so here we're looking at a breakdown of the percentages of what we know about the genome. So as we knew at the end of 2003, only 2% of our genome actually contains genes that code for proteins. So that's what that 2% coding means, is that only 2% of it actually codes for proteins or contains genes. But since 2003, we've come to understand that 10% of our DNA is essential. So that includes that 2% of coding regions, but there's also another 8% of DNA that doesn't necessarily code for a gene, but is essential regions of the DNA. So that means that we've gone in and we've done research where we've kind of removed those sections of DNA and then see how the organism is able to live. And so we have concluded that an extra 8% of that DNA that doesn't necessarily code for genes is essential information in order for us to live. So that might contain regulatory elements or things like that that allow the genes to actually function. Next, we have about 10% of our DNA are introns. So when you look at genes, they contain both exons and introns. Exons being the pieces of the DNA that are actually going to go towards making a protein, and the introns are pieces that are going to be removed before the actual protein is made. And so 10% of that is introns, and while it is generally characterized as junk DNA, the introns are junk DNA, I would argue that when you start learning more about alternative splicing as we move on with these videos, you will start to recognize that introns have a lot of evolutionary value. So they may go on to create new genes that have different functions and allow us to evolve and adapt to our environment best. So while it's frequently claimed that 10% of introns are junk, I would argue that they may have a function that we do not know of, and scientists generally like to claim that if we don't know what it is, it must be junk. But just because we don't know what something is doesn't mean that it might not have a function. So that sort of brings us to our 25% of our DNA has an unknown function. So we have no idea what this 25% does. Maybe it has a function, maybe it doesn't have a function. And this again is frequently classified as junk DNA because we don't know what it is. And so I would again argue that that is a huge amount of human hubris to argue that just because we don't know what 25% of it does, it must be junk. Whereas we know that we discover things that we think are junk that eventually we find out maybe it does have a function. So if we could think of like our appendix as a good example of that. So we've always for like so many years said that the appendix is useless. You can live a totally normal life if you have your appendix removed, which is correct. But we have also kind of discovered through research that perhaps the appendix has some sort of immune function. Maybe it stores immune cells, and while you don't need it, it might be beneficial to have it. And so we definitely have learned throughout history that people like to say, if they don't know what it is, that it's useless or it's junk. But as history continues to show us, it generally does have a function. So even though we don't necessarily know it, maybe we're not aware of its function right now, it doesn't necessarily mean that it will never have a function that we will discover. And so that brings us to the last portion, the biggest portion of our genome, which is 55% transposons and viruses. So if you think back to the first video that I filmed, we were talking about viruses and how viruses actually insert their DNA into their host. So if you've kind of lived about in your life, and all of your ancestors have lived about in their life, they've all been in contact with viruses that are injecting their DNA and integrating their DNA into ours. And so we believe that's where a lot of the DNA comes from. But there's also, from that 55%, some of those are transposable elements. So let's get a little bit into what are transposons and transposable elements. So transposons are also known as transposable elements or jumping genes. So if you see any of these three, they're referring to the same thing. So what are transposons or transposable elements or jumping genes? So these are regions of the DNA that are actually capable of copying themselves and then pasting themselves into a random spot within the genome. So they're called jumping genes because they copy themselves and then they jump to another spot within the DNA. This was discovered by Barbara McClintock in the 40s, where she was studying corn, and she saw that they had a really weird pattern in their coloring. So they had some spots within an individual kernel that were yellow and some spots that were different colored. So she wanted to investigate what was happening there that was causing that color change. And through her investigations, she discovered that this was caused by these transposable elements. So she found that where you saw the yellow colored portion of the kernel, that was where the gene associated with pigment in the corn had that transposable element inside of it. And so that changed the corn pigment in order to be yellow. Whereas when the gene for the pigment did not have the transposable element, it was a different color, like a red or a brown. And so she found that the presence of this transposable element within the pigment gene in the corn was causing the mosaic color within the corn kernels. We believe that these jumping genes or these transposons are responsible for some of the increased size of genomes. So when we look at a rice genome compared to a barley genome, when we're just comparing a few genes together, we see that the genes for the rice are very closely clustered together. And so each gene is kind of in sequence together in the genome. Whereas with the barley, there's large regions of spaces between each gene. And these are all transposable elements. So we really believe that that is part of what has caused the huge increase in genome size from different genuses or different species. So we have found that about 50% of the human genome contains these transposable elements. And then we look at the genes contained within the human genome. We have found that about 70 to 80% of our genes contain transposable elements. So some of those will go on to cause some sort of disease because they now are going to affect the way a protein is going to be expressed, and that's going to affect a whole slew of processes that happen within our cells. But many of these transposable elements actually have no effect, and they've really evolved over time to not jump into places that are going to have an effect on the organism. So we say that these are jumping into safe spaces. So what does that mean? So here, when we're looking at a gene, this kind of a whole gene here, and we see in green, we have all of our exons. And so I can mark here in red the different transposable elements. This is just a made up gene and made up exons. So we see that these transposable elements fall not in the exons of this gene. And so that means that that is not going to affect the actual protein that is made from this gene. And so these are the safe spaces that transposable elements have learned to fall into to not affect the host organism, or just to not affect the genome that it's jumping around into. Because if it's going to cause a really big disease in that person, because it jumped into a very vital gene in a very vital spot, that's not going to be good for the transposable elements either, because now they're going to die off. And so through evolution, only the transposable elements that jump into these safe spaces are going to remain, because if you have a really bad disease, you are much less likely to pass on your genes to the next generation. You're much less likely to live to the point where you're able to have offspring. And so through that evolution of the benefit of having these jumping genes only jump into these safe spaces, that's essentially what we've seen, where now these transposons or these jumping genes really don't jump into vital information. They mainly jump into these safe spaces. But every once in a while, we do see these transposable elements jumping into vital areas, and that can lead to diseases that we see. We've also evolved ways in order to inactivate many of these transposable elements and prevent them from actually jumping into a new space, because it's really a big problem if they start jumping around everywhere and changing the genome that we have, especially when it goes in and changes essential genes that we need. And so we've evolved a way in order to inactivate many of these transposable elements, and many transposable elements themselves have gained mutations that now do not allow it to jump from place to place. So that concludes our video on the Human Genome Project and transposable elements. If you had any difficulty understanding any of the content presented in this or any other video, or anything related to biology, please visit biologybean.com, where you can access all of your tutoring needs. And please don't forget to comment, like, and subscribe.